I feel it's time to jump into our next past paper, where here we have CAPE Unit 1, Paper 2, Biology, June 2015. Let me get started one time. Alright, so you look at question 1. Table 1 summarizes the results of an experiment comparing the effect of temperature on two protease enzymes that break down proteins. One from a mammal that lives in the tropics and the other from a prokaryote living in hot springs. Remember, prokaryote is a very simple unicellular organism that don't really have a true nucleus. The enzyme activity is expressed as a percentage of its maximum efficiency. So if we look here, we could see we manipulated variable, that's the temperature, and that is going from 0 to 90. And we responding variable, which is the maximum efficiency. And we can see for the prokaryote, the maximum, at least the point at which the maximum is occurring, the optimum temperature is going to be 70 for the prokaryote. Right, you see we have 100% efficiency there, whereas the percentage maximum efficiency for the mammalian protease is going to be around 40. Right, so it's going to be around 40, and this is because it's 95% maximum efficiency here. So I mean that's typical of a mammal, because usually a mammal would be thriving in in temperatures that this fluctuates a little bit, and the homeothermic, so the body temperature is around 37, at least for humans. Now for a prokaryote, they say this thing living in the hot springs, so of course the environment is going to have to match the temperature for the enzymes that help it function. So this is why this is a lot higher, which is 70, than the mammal one, which is around 40. So the question here is asking me to plot the data on a graph. So they say on figure 1, plot the data for the mammalian protease as a line graph. So here you're seeing what the graph would look like, and you'll see this is the plotted points here in red. And your marks here will just be correctly plotting the points, making sure you connect them as in the manner of a line graph, and of course labeling the thing, because you have one as prokaryote protease and one as mammalian. And again, what you're seeing here is the maximum efficiency for the mammalian one is about 40, and the maximum efficiency for the prokaryotic one is about 70 from what you're seeing here. And the graph, also wanted it to mark that off, right? So not a hard thing at all. You just plot the graph. You don't have to do up the grid yourself. So the question here is now, using arrows labeled P1 and P2 indicate on the x-axis of figure 1 the temperature at which each protease enzyme functions at its maximum efficiency. So P1 will be the mammalian one, that's 40, and P2 will be 70, which is what we just mark off here, and that would be for the prokaryotic one. Right, everything we just talked about. And they say what accounts for any difference. So the mammal's optimum temperature is determined by its body temperature, while the higher prokaryotic the higher prokaryotic optimum temperature is determined by its environment. Because the mammal is homeothermic, as they regulate their body temperature. The prokaryote doesn't. And so their enzyme will be more determined by what temperatures that it's exposed to, in this case the hot spring. So the maximum efficiency of the enzyme would occur at the optimum temperatures, but different temperatures in this case, because it's two different types of organisms. Alright, part 3. Enzymes are mainly globular proteins. With reference to the levels of protein organization, explain the effect of temperature on the prokaryote protease between 75 and 90 degrees Celsius. So let me look at the table here. So you can see here between 70 to 90 degrees Celsius, and right above that, this thing going down, the efficiency going down, and you could also see this on the graph. Right around here you see plummeting right down. So why does the maximum efficiency of the enzyme decrease? Well, this is what we have to understand here. Well, on a molecular level, if you increase the temperature of those molecules, they're gonna gain kinetic energy. What's up when something gains kinetic energy? They're gonna start vibrating. And when they vibrate more and more and more, well, they vibrate before, but they'll vibrate more. And when they vibrate more and more, their bonds will start to break. The weakest ones first, that'll be like the Van der Waals forces, the hydrogen bonds. Those bonds will break. And those bonds, remember, are holding the molecule together and holding the structure of the protein together. So the enzyme has a tertiary structure, which is, you could think of it as a three-dimensional structure held together by bonds and holding together all these polypeptide chains that bonded together and shape like alpha helices and beta pleated sheets. All you know the deal. So anyway, so what happens now when this tertiary structure starts to unravel? Well, this would change the shape of the active site where the substrate is supposed to fit. So if this is your substrate here, and there's your enzyme here, and there's your active site here, and usually this will fit, like a hand in a glove. But if you was to actually change the structure of the enzyme, like it was a heater, the vibrate bonds at the break, the substrate now would not be able to fit or would not be able to completely bind to the active site. So heating it would change the shape of the active site, and this would prevent the substrate from completely binding, thus decreasing activity. At a point now, the enzyme is going to be irreversibly damaged, and thus all enzyme activity will stop. And remember, that's called denaturation. B. Figure 2 is an electron micrograph of a mammalian liver cell. Here we have a bunch of organelles, and the first question is actually asking me to label what is 1, 2, and 3. Well, 1 is the mitochondria, and you could tell from the little folds on the inside, right, if you look closely enough, and you could also tell from this slightly elongated shape, kind of like a bean, and that's the mitochondrion, which is used to release ATP. 2 is the ER, and you can't really tell if it's the smooth or the rough, because you can't really see the ribosomal structures from this point, so you could get away with just saying ER, or if you want to feel bright, endoplasmic reticulum. And 3, which is usually the most prominent structure in a cell, this is the nucleus, which holds the DNA of the cell. Right, so B part 1, you'll see that we have identified mitochondria, ER, nucleus. Now part 2 is saying if the actual size of the structure labeled Y is 1.5 micrometers, calculate the magnification of the electron micrograph in figure 2. So this is the structure Y here. Now you can't do this on a computer screen, but if you have a physical copy of the paper, if it was to measure end to end of Y, this is actually 11 millimeters. So if we was to take now the size of the drawing, which is 11 millimeters, or the size of the image, and convert it to micrometers, we get 11,000. Now the magnification would be equal to the size of the image divided by the size of the actual object. So this would be 11,000 micrometers divided by 1.5, and you get 7,333. Now of course the marks here just give a little leeway, so if your ruler bad, or if you can't really see good, then I think this give you a range here between 7,000 to 8,000 times, and you should be alright if you're within that range. The last question here suggests a reason which may account for the extensive network of membranes seen in this type of animal cell. Well, remember, there's a liver cell, and this is filled with a whole set of folds of ER, right? So remember, that is used to synthesize proteins, and the smooth ER is used to synthesize lipids. It is also used for the transport of materials, but because it's the liver, it also used for storage of things like glycogen. So good answers would be allows for efficient transport of metabolic products such as bile salts, like to emulsify fats, or lipids, or allows for protein and lipid synthesis by the rough and the smooth ER respectively. Basically any answer that would make sense in context of synthesis of materials or storage of materials should be okay. Alright, so let me look at the next question. Question 2, God always is a cell division question. Oh, God is a meiosis 1, 2. Alright, fortunately, it not bad. So you look at here. Figure 3 is a diagram of a cell with 4 chromosomes. The haploid number is 2 during the process of meiosis. So you see 4 chromosomes here. 1, 2, 3, 4. And now what you're seeing here, because it's meiosis, now you'll notice that there's a chiasma forming here. Chiasma is basically a region at which these 2 chromatids are going to exchange information. They're going to cross over. And that doesn't happen in mitosis, right? So they basically pair up here, and you can see that there's points of exchange and crossing over. Anyway, so at this point here where they're not aligned really, so they're not really aligned at the equator of the cell, and of course they're not being pulled apart yet. At this point they're just kind of hugging up. Well, the name at this point is prophase 1, right? The first prophase. And this is because, you know, it still have 4 chromosomes in here with all the chromatids intact. Now in the boxes below, make label sketches to illustrate 2 key stages of meiosis in this cell, and write the names of the stages on the lines provided below in boxes. So the next 2, so remember, if you remember PMAT, or IPMAT if you want to include interphase, but interphase is not really one of the steps. That's just like a precursor to the steps. You know, you have prophase, metaphase, anaphase, telophase. So how I like to remember it, and of course you'll have a second round of these in meiosis too. But how I like to remember it is prophase is like prom. You know, like when you go into your prom and you have to dress up and you have to, you know, you're hugging up and thing. Basically what you're seeing here. Right, so everybody just kind of pair up and dress up, you know, and they have on they take the chromatin threads and they make a nice chromosome dress out of it, you know. Metaphase is where they will start to meet in the middle, right, where everybody will start to align in the middle of the cell, kind of like, you know, everybody meeting up during graduation. Anaphase is when, you know, sometimes you have a fight at the graduation and they get pulled apart. Everybody angry with each other. Angry anaphase. So this is when things start to pull apart. And well, after a point, you go home and they call each other on the telephone, but you're no longer in the same place. So telophase, T, is where the cell division actually starts to happen, where cytoplasm and so on starts to split, and you get cytokinesis. And the nucleus splits, and you know, you're basically one cell ready to turn into 2 cells. So prophase, metaphase, anaphase, telophase. The next 2 would be metaphase and anaphase. So let me look at those steps there. So here we have metaphase 1 and anaphase 1. So I say metaphase is where they would meet in the middle during prom, or they meet up, and you can see that there's still chiasma forming here. Right, plural is chiasmata, and this is where you'll start to get some genetic exchange, some crossing over. So these chromosomes, these tetrads, start to line up, and anaphase 1, remember I said that's like when everybody angry with each other, had a little fight, and this is where they start to get pulled apart by the spindle fibers that you're seeing here. And so when they get pulled apart, they will move from the equator of the cell to the poles of the cell, and right after that is where you'll start to get telophase happening and cytokinesis, which we don't have to talk about here. But the homologous chromosomes split up at this point. So once again, that's where you'll be drawing there, and I'll just put it up on the screen here briefly so you'll know what you have to do. Of course, you name the stages there: metaphase 1, anaphase 1, and they try to label the things that are going on. So the question here now asks, explain how the key stages identified in A1 and A2 contribute to genetic variation. So once again, the 3 stages was prophase 1, anaphase 1, and metaphase 1 and anaphase 1. You could talk about any ones you want. But what's happening here, as I said, you would have chiasmata, or regions of crossing over occurring between the chromatids or the tetrads in prophase 1. And that there is gonna make sure that some DNA from one goes to the next one and vice versa. And so what will happen is that the daughter cells will not be clones of the parent cell. They will all have DNA or genes exchange between each other. Now, in anaphase 1, homologous chromosomes undergo independent assortment, and due to the random alignment, what you'll also have is many different arrangements of alleles. So because these chiasmata could form at any point, so if I was to look back at this diagram here, you'll see that where we have chiasmata here, but they could occur almost anywhere, right? And in different cells it would occur at other points. So you get plenty different possibilities of exchanges of DNA and little differences in the daughter cells, which will eventually be in the gametes. Yeah, so there can be many different arrangements of alleles. So it's 3 marks. Once you make about 3 points, you should be fine, but you want to focus on things like crossing over, independent assortment, random alignment, things like that. The fact that they could get a large possibility of alleles in these daughter cells. Okay, here we have a kind of tough one. Explain how substitution and insertion result in genetic variation. Discuss how gene mutation causes sickle cell anemia. Alright, so substitution is like if I have let me say I have this, right? Let me say I have this set of nucleotides and one of these get replaced. So let me say that this thymine gets replaced for an adenine. And instead of GTG, you end up with GAG. So that's a substitution or a point mutation, because we have basically this will form an entirely different amino acid. And we end up with a problem, because when we need to make the RNA equivalent to this, right? So remember, G, this pair with G, this pair with C, and T, this pair with A, and G is paired with C, and the RNA equivalent for this here, G is paired with C, and A for RNA wouldn't pair with a T, it would pair with a U, and G would pair with a C. So what we end up is the translation of an entirely different protein from these base pairs. Right? We don't need to talk about all of that, but the point is here is that the blueprint is wrong. The instructions written wrong. So the building, right? The construction of the building gonna come out wrong. So substitution occurs when a nucleotide is replaced with another, like A instead of T, for example, C instead of G. And thus a change in base pairs occur. This results in a different amino acid sequence, and a different protein can be made. Right? So not always, but most likely. Insertion, on the other hand, is where you end up with the addition of a nucleotide or base pair in the DNA code. And what will happen there is that because bases are read 3 at a time, this is going to cause a frame shift. And a frame shift means that the whole thing is going to get thrown off. So if I had A, T, C, and somehow a G get added in there, A, G, T, C, well, what's gonna happen here is that instead of whatever is gonna is gonna be made from ATC, is gonna read from AGT, C whatever, right? So it's gonna cause a frame shift, and that could actually affect the entire protein structure. So these are generally kind of a little more dangerous. But here they're talking specifically about sickle cell anemia. And sickle cell anemia, well, that is actually caused by a substitution, where you end up with an A being replaced with a T in one instance. So what happens there? Well, everything I just said. So as a result, the polypeptide chain in the hemoglobin is changed. So instead of the protein GLU, you're going to end up with instead of the amino acid GLU, you're going to end up with an acid called VAL, right? Valine. And you don't need to put all that here, but you end up with a whole different thing being formed. And if you have a different amino acid, the way in which those amino acids could be bonded, the way in which the 3D structure of the hemoglobin is would change. So it's kind of like a cascade effect, if you could understand what I mean. If the thing wrong from lower down, the thing gonna be wrong from higher up, most likely. So the structure of the hemoglobin changes, and the red blood cell, instead of having a biconcave shape, becomes sickle shape instead. Their oxygen carrying capacity is reduced. So what you want to do is you want to explain what is substitution, explain what is insertion, state how sickle cell anemia occurs, how it affects protein shape, and also how it affects protein function. Alright, question 3. Table 2 is an incomplete comparison of some features of a human ovum with some features of a spermatozoon. Alright, so remember, ovum is a female gamete, spermatozoon is a male gamete. And this is a pretty easy table to fill out. So the first feature that you're looking at here is the size. And the size here of a spermatozoon is over the ovum. It can't be seen with the naked eye. Well, you know, you can't really see sperm with the naked eye, so it cannot be seen. And it's about 0.05 millimeters long. That's just extra information. You need to write that. The ovum, so the shape of that tip, you know, if you see it is more round or spherical. Spermatozoon has a more elongated shape, which, you know, it's kind of like it's stretched out because it have a head and a tail and a neck and a middle piece. And that's common to the overall structure here. Whereas the ovum is not really differentiated or divided into distinct regions. As I say, it's more like a sphere with several layers. Where the follicle cell, the corona radiata and thing. The sperm cell, however, is actually quite differentiated here. Again, you have a head, a neck, a middle piece, and a tail. Motility, that's basically the ability to move by itself. Well, the egg can't. Usually they are brushed, is carried by the brushing motions of villi in the fallopian tubes, but the sperm cell can. It is motile, it can't swim, it has a flagellum. Food reserves. So the sperm cell doesn't have any food reserve. They're not really expected to live very long. It really just had to, you know, get out there and fertilize the egg. The egg, however, is a lot larger. It has a lot more cytoplasm in it, and it needs more long-term nourishment. It has to basically be there for at least a few days. So there's some food reserves in there. Metabolic activity. Well, the sperm cell is really inactive in that it does not absorb or release any substances. Whereas the egg or the ovum, it does. As substances are absorbed and released. I say is mainly because, you know, it has food reserves, and then it releases certain chemicals that causes capacitation in the sperm cell, so it could locate the egg cell and so on. Right, so that was 6 marks, and it's basically 1 mark per thing there. Right, part 2, with reference to a specific function of the human ovum, discuss how its structure is suited to the stated function. Well, really, the function of the ovum is to, you know, get fertilized by the sperm cell, or to basically allow reception or entry of the sperm cell. So how does it do this? Well, it has an outer layer, and that contains cell surface proteins. And what that would do eventually is that it would form a fertilization membrane that will only allow entry of one sperm cell. So you can't have all of them coming in one time. So once it gets fertilized, that will happen. And in terms of the structure of it, we know the egg cell is a lot larger, and that facilitates easier contact with the sperm cell. Once you use a little bit of common sense, you should be alright with that one. Alright, so figure 4 is a diagram of a cross-section of a fertilized ovule of a flowering plant. And here we can see the pollen tube, and this would have been moving down the style to come into the ovary. And here we could see all our friends here that, you know, that the pollen nuclei would have moved into the male nuclei. And it already happened because you could see at Y there's this is a triploid nucleus, so that means there's 3 sets of chromosomes here. And where I marked X here, this would have been where the egg cell was, which has been fertilized, so there's a diploid now. So there's a zygote. So X is the fertilized egg cell. So this will indicate using an arrow labeled X. So when I put an arrow display there, the structure which should develop into a radicular plumule, that would be the fertilized egg cell, which would become the embryo. Identify the structure labeled Y and explain its role in the developing embryo. Well, this is your endosperm nucleus, and which is a triploid. This develops into the endosperm, and what the endosperm acts is like a reservoir or food reserve for the embryo. Is basically like the placenta for the little plant baby that are gonna form. Last one here in this question. An orange is described as a fleshy fruit. Name the part of a flowering plant which develops into a fleshy fruit after the ovule has been fertilized. Comment on the importance of the fruit, such as an orange, in the life cycle of a flowering plant. Well, first of all, the part that becomes the fruit is the ovary, and the flesh of the fruit is actually the pericarp. Right? So you could put that too. And well, what is the importance of the fruit? Well, the fruit is there for multiple reasons, but the two main ones we could talk about is that it's there to actually prevent mechanical damage of the seeds, because it acts like a casing, right? That holds the seeds in place, also prevents the seeds from drying out or desiccation. The fruit is also necessary for not all, but animals that disperse seeds. Basically, they will eat the fruit, and then the tester or the seed coat would digest. It will get broken down in their digestive system, and then they will defecate the seeds, basically plant them somewhere in soil, and basically these seeds could be transported and dispersed far away from its parent plant, and that has a lot of advantages, which you don't get into here. Question 4. With the aid of an annotated diagram, describe the structure of phospholipids. Alright, so I'm gonna put up a diagram here so you'll see what to draw. Now the diagram that I have there is actually including the molecular structure of it. You don't need to draw that to get all your marks here. You could draw a simplified version of it where you just draw the phosphate head, the fatty acid tails, and a glycerol molecule connecting the two. There's no need to state the individual atoms and the bonds and so on. Once you draw something basic in this case, you should be fine. Now if they said describe the molecular structure of it, or draw the molecular structure, then you would want to draw your your phosphorus and your oxygens and your carbons and things like that. In this case here is only 4 marks, where most of it is actually there for the description. So that's that. So let's look at the description now. So the phospholipids, right? So phospholipids are what you call amphipathic, which means that they consist of hydrophilic or water-loving phosphate heads and hydrophobic or water-hating fatty acid tails, because they're very oily and they repel water. So they hydrophobic in that sense. And in your annotations, well, you would have seen it there, but you kind of describe what the phosphate head is made up of, which is a phosphate atom kind of bonded to oxygens, and fatty acid tails are long strings of carbon, and there may be like little kinks right in the fatty acid tails. So these are connected by glycerol molecules and true bonds called ester bonds. Now we don't need to go into any more detail here. Once you have a little description down here and some good annotations and labels here, you should be able to get your 4 marks. Part 2. With reference to the fluid mosaic model, discuss the role of phospholipids and proteins in cell membrane functioning. Right, so you'll be lucky if you get a question like this. This is pretty easy. The fluid mosaic model basically says that you have a sea of lipids or a sea of phospholipids with proteins floating throughout. So they're not static, they actually move throughout the sea. through the phospholipids. And I like to think of it like the proteins are like icebergs floating in the ocean, and again, those are not in one place, they sort of slowly move around. Now, what are the role of each? So the phospholipids role is to form a barrier that would separate the cell interior from the exterior, the interior being the cytosol. And what the phospholipids would do, and because it has that hydrophobic internal part, right, especially if it's a bilayer, what that does is that prevents any larger polar molecules, even water, from passing through. And it only facilitates movement of non-polar molecules like gases, carbon dioxide, but larger molecules like RNA, like glucose, those won't really be able to pass through the phospholipids, which is kind of like, think of it like a fence. They wouldn't really be able to pass through those little gaps. Now, however, what the proteins are there for, especially if they are intrinsic or integral proteins, which means that they span the entire width of the bilayer, those can act as either channels or carriers. Channels are just like little tunnels that larger molecules could pass through, like glucose. Sometimes they can be specific. Carrier proteins, on the other hand, they act like gates that you need a little key, so they have little binding sites to allow movement of ions or larger particles. A good example is moving sodium and potassium through pumps in the neurons. Those are carrier proteins. They require, the sodium and the potassium have specific binding sites that allow, facilitate movement in and out of the cell. Proteins can also assist with cell recognition and signaling, and remember, proteins are also, enzymes are also proteins. So proteins that are enzymes carry out metabolic reactions. Now the proteins some of them would have glycolipids attached to them. They would have other molecular structures attached to them, and those could be used for identifying certain parts of the cell, and as the cell recognition and signaling. Basically, once you have a basic description was the fluid mosaic model, a good description of the role of the phospholipids, a good description of the roles of proteins, you'll get your 5 marks. Alright, so they say, using a generalized structure for amino acids, outline the process of the formation of a peptide bond between two amino acids. Explain, using an example, how amino acids differ from each other. Okay, so the basic structure for amino acid, if I was to draw two of them, is that you have a carbon, and this would be connected to a nitrogen here, and you would have a hydrogen over here, and then basically NH2 there, and you would have a residue group, basically an R group, which will be different for each amino acid, which actually explains the next part. A hydrogen atom here, and a carboxyl group here. So this would be C, right, oxygen, and OH here. And if you was to do this again over here, basically the same thing, and I just draw in one part of the second amino acid, but you'll notice here when this combines, well, this is H2O, and this here would be a condensation reaction. So this bond is going to form between the carbon and the nitrogen here, and this is what's going to be a peptide bond, and this water here is going to be lost as a result. So what I'm going to do is I'll post a picture of what would be the ideal drawing for this answer, and you'll see. Now, to answer the question, what basically makes amino acids different from each other are these R groups, which are called residual groups. So amino acids vary from each other based on the R group attached, and these R groups are, they can vary in size, polarity. Some of them are neutral, some of them contain aromatic rings. They have all different types of bonds. And that's how you get all these different types of amino acids. This is what would separate, if you use the sickle cell anemia example, this is what would separate the GLU from the VAL amino acid. Okay, so we have a kind of tough one here. Question 5. Explain what is meant by the term gene, this is the easy part, and outline the principles of using restriction enzymes to cut a gene from a length of DNA. And you don't need to involve all the steps of recombinant DNA technology. You just have to just explain basically restriction enzymes. So we kind of get away a little bit. So to define a gene, you could define it in two ways. You could say it's a basic unit of inheritance that determines traits or characteristics. You could also define it as a segment of DNA on a chromosome, and that segment of DNA would code for a sequence or a specific sequence of amino acids or a protein. Now, what a restriction enzyme is an easy way to think of what a restriction enzyme is to think of it like a piece of scissors that cuts DNA. And what a restriction enzyme is, or where it comes from, is that it's usually produced by bacteria. And they are given names based on the bacteria that produce them. So, for example, you have one called Echo R1. And what they do is they help the bacteria's defense against viral DNA. So when viruses invade bacteria, the bacteria produces restriction enzymes, and that helps to cut out the viral DNA, basically to make it inert. And yeah, that's basically the point of it. And why they call restriction enzymes is because it actually restricts the virus from acting further, or the virus from actually taking over the bacteria. So that's why it's called a restriction enzyme. So how it does this is used to cut across DNA strands at specific sites, and they would have specific nucleotide sequences. So you have a BAM H1 Echo R1. If you look up those things or you see it in the textbook, they are specific sequences, and they typically have to be what you call palindromic. Palindromic means that it can be read forward and backward. In this case here, 5' to 3' or 3' to 5', in the same way. And your palindromic words too. An example of a palindromic word is Bob. B-O-B could be spelled the same way forward and backward. And another one is race car, which again could be spelled the same way forward and backward. In this case here, you would have DNA sequences, nucleotide sequences that would be the same forward and backward. I'll just give you an example. It would be like ATTA, right? Same way. So they are usually made by bacteria to cut out viral DNA. Think of restriction enzymes as DNA scissors. Now, usually when they make these cuts, they will leave free unpaired ends. And these unpaired ends are called sticky ends, and these sticky ends are used for attachment to easy attachment to other sequences. I'll put up a little diagram here just so you'll see what I'm talking about. Alright, so B, gene therapy is the principle of genetic engineering in the treatment of diseases. Give a brief description of one human disease for which the use of somatic gene therapy has been shown to be effective. Now, the two diseases that you might be familiar with here is cystic fibrosis and SCID. But the one that you most likely do with the teacher is cystic fibrosis. That's the one that's shown here. So the example is cystic fibrosis, and that's caused by a substitution mutation, which means that one nucleotide was off. One was replaced by one that not supposed to be there. Now what happened there is that this code for an entirely different protein. Now usually you'd have a channel protein called CFTR, and that protein allows the passage of chlorides and other ions through it. And usually you want those chlorides to move through that channel, because if too much chlorides start to accumulate in an area, it'll cause a salt imbalance and a build-up of mucus because of inflammation. Now part 2, and this is 8 marks, which, the questions like this could be kind of scary because they are multi-marks. Discuss 4 reasons why somatic gene therapy, despite its potential, is still not used as an effective treatment for human diseases. So somatic gene therapy is where they would try to insert the what we could say is the fixed DNA, the good DNA, the repaired DNA, or try to, it kind of like when you're fixing a computer bug, they try to insert it into into a specific target cell. So not into the gametes, right? So in this case they're talking about cystic fibrosis. They actually use a little nasal spray to deliver the fixed DNA through little lipid droplets. Now sometimes they use viruses to transport the DNA, and why viruses is because, well, viruses are real good at invading your cells. So what better way to actually try to get it into your cells? So they might put the viruses into your lungs, and you inhale it, it goes into the cells, penetrates the cell membrane, and delivers that little bug fix, that little fixed DNA into you. Now, of course, we have a problem, because if we're using viruses, well, we know viruses, it can't be good. We have a lot of risk associated with it. So viral vectors may be used to transport the new DNA, but it could cause inflammatory responses or severe immune responses, which would cause a lot of discomfort or even debilitation of the patient, especially if they immunocompromised or they're old. So the virus may also become a pathogen. So you don't know if the virus might actually just regain its abilities while inside the patient. There's a lot that we don't know. There's a lot of research still to be done. So there's always a high risk of that happening because they're using these viral vectors to transport this new DNA. The next thing here is that at this current point, somatic gene therapy is limited to diseases caused by only a single gene mutation. So if you have a disease that affects multiple genes, or is caused by the affectation of multiple genes, also it's multifactorial, you cannot treat it effectively in this, because you have an actual limitation to the delivery of this. Is expensive, so because it usually requires therapeutic treatments over the course of the patient's life. So and then you have the cost of research and thing too. The other thing here is that the effects may not be permanent, because the repaired cells might actually die instead of proliferate. What you want to depend on is that those repaired cells, that they proliferate, that they divide and they form new good ones. So that's not really likely to happen. They might just ultimately die and be replaced by new cells. And the thing is, the virus, we can't really tell the virus which cells to invade. Not all of them might be bad. So they may not always target the correct set of tissues to deliver the DNA. They might end up delivering it to other cells instead. And it basically is inefficient in that way. So these are 4 reasons that you could use. Other ones would include, some people, I don't know which people, just think it unethical. You're playing God. I mean, there's actually legit reasons to argue here. Basically, if you can't think of nothing, you could put that as one, in case you see this question again. Alright, question 6. Explain 2 mechanisms for the promotion of cross-fertilization in plants, or basically what you call outbreeding. So you have plenty of different ones you could use here, but I use 2 kind of simple ones. It's 4 marks, so you want to have at least a good description of each one that you have. So the first one here I have is dioecious plants. So dioecious, the word dioecious means 2 houses, and what that basically means is that males in one house, females in the next house, males in one plant, females in the next plant. And what dioecious plants contain is male and female flowers on separate plants, thus preventing cell fertilization. So you would have a male plant and a female plant, right? Kind of like cannabis. 2. protandry and protogeny. Protandry means that the male parts are formed first. Protogeny means that the female parts are formed first. These ensure that the male and female reproductive organs, which are the stamens and the pistils, do not open or mature at the same time. And so I like to think of it like this. Protandry is basically the man wake up first, the woman still sleeping. Protogeny, the woman wake up first, the man still sleeping. Nothing could happen. Right, so nothing can happen there because both they're not active at the same time. They don't open or mature at the same time. It have three others here. You could use self-incompatibility, which is where the plant would produce pollen that would not be viable with the female with the pistil. So the pollen would not produce a pollen tube if it was to land on the same stigma of that flower. It would have to look for a flower with totally different alleles. It's basically like opposites attract, if you know what I mean. It don't want to be with the same flower. Male sterility, which means that if you have a plant with male parts and the pollen would not be viable at all, it might not produce the pollen at all. And heterostyly is where you would have two different types of female parts. I'll put up a little diagram just so you could see what I'm talking about, but you'll find more of it in your textbook. Alright, so this part 2. Evaluate the genetic consequences of self-fertilization, also called inbreeding, and cross-fertilization or outbreeding in plants. Or just to explain, self-fertilized plants are those that are self-pollinated or inbred. Cross-fertilized plants are those that are cross-pollinated or outbred. Basically, you need two different plants of the same species. It's not the same as, I should just note here, self-pollination is not the same as asexual reproduction because you still have gametes involved, right? Anyway, so self-fertilization, which again is inbreeding, you only have basically the one parent or the one flower that fertilizes itself or pollinates itself. Well, because now you have is the same organism, this would reduce genetic variation in flower populations and thus leads to reduced vigor. And what that means is that they're less likely to overcome any selective pressures. So if you have a bunch of self-pollinated flowers, what would happen is that if you have a pest that would target one of the plants, or you have a disease or a pathogen that could affect one of the plants or one of the flowers, it could potentially affect all of them. And you have less biodiversity, so to speak. Now, you would only really do self-pollination if the environment or the ecosystem is stable enough, or if it's a person who want, they want to sell some flowers and they have some bright petal flowers, and they would self-pollinate them because they want to keep the advantageous alleles and traits, which is the bright flowers, to pass down to the offspring. Cross-fertilization, however, is the opposite in terms of these effects. They would increase genetic variation and vigor in a population. They're more likely to overcome selective pressures. So if you have a pathogen, it might affect a few, but it might not affect all. So you'd get at least some surviving. And that would lead to greater evolutionary potential and a chance for speciation. One species of flower branching off into many different species, which is always good, I guess, for life and for the earth. And the last question of this paper, starting with contact of sperm cells with follicle cells of the oocyte, the secondary oocyte, which is released during ovulation, outline the process of fertilization in the human reproductive system. I would say any 4 of these points here should be fine to get 4 marks. What would happen is that the sperm undergoes capacitation. That's basically when it the acrosome activates within it and it starts to swim faster towards the egg. Basically when it knows the egg is near, when it gets the little chemical signals from the egg, the acrosome now, which is at the head of the sperm, releases enzymes. These enzymes now digest a pathway through the follicle cells and the zona pellucida in the secondary oocyte. So basically sperm, you could think it kind of like had to digest or eat its way through, not eat, but digest its way through the to get to the female nucleus. Sperm cell receptors bind to the zona pellucida and the sperm nucleus enters the secondary oocyte. So the sperm nucleus basically exits the head of the sperm and goes towards the into the oocyte. Well, what would happen now is the zona pellucida will become impermeable to other sperm cells. It becomes a fertilization membrane and it prevents them from binding. So it would only allow this one sperm cell to enter due to this. This is after something called the cortical reaction. The secondary oocyte completes meiosis 2. There's a whole story in itself, but basically what happens is that the secondary oocyte only finishes up meiosis 2 when it is fertilized, and then it becomes an ovum. And when that happens, it basically divides and takes out 23 chromosomes into a polar body. Again, there's a whole different story, right? I have other past papers with this, but it becomes an ovum at that point, and then from the ovum, right after it becomes an ovum, the sperm and the oocyte nuclei fuse to become a zygote. I say that there's a whole story by itself. But yeah, but so if you just have 4 of these, you don't even have to put this last part. If you have 4 of these and you just have the 2 nuclei fuse to form a zygote, it should be fine. Alright, all you. So that is it there. Enjoy your day. Laters.